Hello and welcome everyone. My name is Adriano Martinelli and I am from IBM Research in Zurich and ETH Zurich. And today I would like to present to you a tool that we developed called ATHENA that can be used to analyze tumor heterogeneity in spatial omics datasets. So tumors are spatially organized heterogeneous ecosystems, as shown here and exemplified here with this ductal carcinoma, and they consist of many different cell types, like normal cell types, different tumor subclones, or immune cells. And all these different cell types are now interacting with each other in this microenvironment. They can compete with each other for nutrients or oxygen, but they can also cooperate with each other and, for example, share certain types of factors that they produce, like proteolytic or angiogenic factors. And what we try to do with ATHENA is to actually capture a few of those characteristics in this microenvironment and quantify them with different methods. Now I would like to quickly talk about these new spatial omics techniques upon which the computational framework of ATHENA is built. So over the last few years it became possible to quantify dozens of proteins or thousands of transcripts in tissue samples. And these techniques are actually preserving the architecture of the tumor ecosystem. And as an output you receive, all these techniques actually produce some kind of a image output in which the pixels in the image represent the abundance of proteins or transcripts. And one of those spatial omics technologies is the imaging mass cytometry technologies, in which a tissue sample is stained with an antibody panel, and then a laser ablates the tissue sample pixel by pixel, and the ablated particles are subjected to a CyTOF measurement, and this results then in these images here on the right hand side that quantify the spatial distribution of the protein abundances. And these types of images, together with some segmentation masks that need to be produced by third-party tools, are then the input to our ATHENA workflow. Based on the images and the cell masks, we can then phenotype the different cells and from that produce a graph representation of the images. This graph representation is then used as an input to our quantification module, where we quantify different aspects of that graph and the heterogeneity that we see in this graph. The quantifications that are resulting from this analysis can then be used by the user for further downstream tasks. So ATHENA provides three different ways to construct a graph. A radius graph, which can be used to model paracrine signaling, a contact graph, which connects cells that are in physical contact and that can be used to model juxtacrine signaling, and a KNN graph representation. Next, we also implemented several entropic scores that are borrowed from ecology and information theory. And in general, these scores are a function of the number of species, the so-called richness, and their relative abundance. So first of all, like how many different cell types are in a sample and what's the distribution of those cell types in the sample. And here we can now see two different examples, one with low entropy on the left hand side and one with high entropy. And if you look here at the bottom on the histogram, both samples have a comparable richness of 21 and 19, meaning that in both samples are 21 different phenotypes or 19 different phenotypes. But the sample on the left hand side, the distribution of these phenotypes is very skewed towards one single phenotype, and this leads to the fact that the Shannon entropy of this sample is much lower than the one on the right hand side, in which the phenotype abundances is more balanced. So in their original definition, these entropic scores result in one single level per sample, because you look at the whole cell type distribution across a sample and then compute the entropy from that. However, we adapted this score a bit to actually result in single cell entropy values by exploiting the graph representation that we constructed at the beginning. And we do that by, for each cell, we look at the neighborhood of this cell and compute the cell type distribution from this neighborhood, which is defined by the graph. And therefore, we are able to compute the single cell entropy values that kind of quantify how homogeneous or how heterogeneous the direct environment of each single cell is. And here you can see 3 examples. On the left-hand side is one in which the environment of the cells is very homogeneous because they are surrounded by the same cell type. And in the example on the right-hand side, the cells are surrounded by many different cell types, and thus the entropy values are much higher for each cell. And this is also then reflected in these histograms at the bottom of the entropy, the single cell entropy values, where in the case where the environment is very homogeneous, so the left-hand side example, the distribution is skewed towards low values, and in the example where the cells are intermixed, the distribution is more skewed towards high values. And this is now also exemplified here with these 3 real-world cores. At the top, again, you see the different phenotypes, and then in the middle, you see each cell colored according to its entropy value, and you can see how so that the more yellow the color, the higher the entropy. You can see how this changes from left to right. And then also if you look at the distribution of the entropy for the single cells, you can see how on the left-hand side the distribution is very much skewed towards very low values, and then on the right-hand side it's more evenly or even skewed towards higher values.